Tooth defects and tooth loss are common clinical problems affecting millions worldwide. Traditional dental treatments, such as fillings, crowns, and implants, have helped restore function and aesthetics, but they do not truly replace the lost living tissue. The dream of regrowing natural teeth capable of sensing, healing, and adapting has become a major focus in oral biomedical research. Recent advances in developmental biology, stem cell science, and biomaterials are converging to make tooth regeneration a realistic goal. In this video, we'll explore the science, strategies, and future prospects of tooth regeneration. To understand tooth regeneration, we must first look at how teeth naturally develop. Tooth morphogenesis proceeds through distinct stages: initiation, bud, cap, and bell. Specialized signaling centers, such as the enamel knot, and structures like Hertwig's epithelial root sheath guide the formation of tooth shape, cusps, and roots. This process is orchestrated by the spatial and temporal expression of key molecular pathways, including BMPs, FGFs, SHH, WNTs, and TNFs. Misregulation of these signals can lead to developmental abnormalities, highlighting the importance of precise control in regenerative strategies. Tooth regeneration is an emerging field aiming to regrow natural living teeth and their supporting tissues using a patient's own stem cells. Unlike artificial restorations, regenerated teeth can sense pain, remodel, and integrate seamlessly with the jaw. The scope of tooth regeneration includes three main clinical objectives: whole tooth regeneration, dental pulp and dentin regeneration, and periodontal regeneration. Each objective targets different tissues and presents unique scientific and clinical challenges. Successful tooth regeneration relies on four interacting components: suitable cell sources, scaffolds to guide tissue formation, a cellular growth factors to direct differentiation and angiogenesis, and an appropriate culture or incubation system. Progress has been made in each area, but no single solution yet reliably reproduces a complete, functional tooth in humans. Combinatory approaches that integrate these pillars are currently the most promising. Stem cells are the foundation of tooth regeneration. Several types are under investigation: induced pluripotent stem cells, iPSCs, mesenchymal stem cells, MSCs, dental-derived stem cells, and endogenous stem cell homing. Each has strengths and limitations. For example, iPSCs offer high differentiation potential but carry tumorigenicity risks, while dental-derived stem cells are naturally predisposed to form dental tissues but are limited by donor availability. Understanding these sources is key to advancing regenerative therapies. Dental tissues provide several types of stem cells with regenerative potential: dental pulp stem cells, dental follicle stem cells, stem cells from human exfoliated deciduous teeth, stem cells from apical papilla, periodontal ligament stem cells, and gingival stem cells. These cells are accessible, suitable for banking, and show high proliferative and multi-lineage differentiation potential. Their compatibility with scaffolds makes them attractive for tissue engineering. Scaffolds are three-dimensional porous biomaterials that provide spatial cues, support cell attachment, enable nutrient transport, and degrade controllably as new tissue forms. They can be natural, like collagen or chitosan, synthetic, such as PLA or PLGA, or composite. The ideal scaffold is biocompatible, matches the mechanical properties of dental tissues, and supports vascularization. Recent advances include 3D printing and bioactive glass composites, which allow for patient-specific designs and improved integration. Growth factors and cytokines orchestrate cell recruitment, proliferation, differentiation, and tissue patterning. Key players include PDGF, TGF-beta, BMPs, VEGF, FGF, IGF, and NGF. These signals act in a highly spatial temporal manner, meaning their effects depend on timing, location, and concentration. Controlled delivery of growth factors is essential for successful regeneration, as it mimics the natural developmental environment. Incubation systems provide the environment for tooth tissue growth. Two-dimensional cultures are useful for basic studies, but lack complexity. Three-dimensional cultures better mimic tissue architecture and cell interactions, essential for studying mineralized tissues. In vivo animal models are necessary for testing vascularization, innervation, and functional integration. Each system has strengths and limitations, and combining them helps bridge the gap between laboratory research and clinical application. Tooth regeneration strategies can be scaffold-based or scaffold-free. Scaffold-based approaches use biomaterials to guide tissue formation, control cell placement, and deliver growth factors. Scaffold-free methods rely on the self-assembly of cells into sheets, spheroids, or strands, preserving natural cell-cell interactions and reducing immune reactions. Each approach has unique advantages and is suited to different clinical scenarios. Scaffold-free constructs include cell sheets, spheroids, and tissue strands. Cell sheets are fabricated using thermoresponsive polymers, preserving cell-cell and cell matrix connections. Spheroids are formed by various methods and support strong cell signaling, vascularization, and differentiation. Tissue strands are created by molding cell suspensions, allowing for linear arrangement and integration of multiple cell types. These constructs show promise for pulp and dentin regeneration, but mechanical stability and vascularization remain challenges. For regenerated tooth tissues to survive and function, they must be rapidly vascularized and innervated. Scaffold-free spheroids and strands show enhanced angiogenic and neurogenic potential due to their 3D architecture and cell signaling. Pre-vascularization strategies, such as including endothelial cells and neurogenic induction protocols, are being developed to improve integration with host tissues. Achieving reliable blood and nerve supply remains a critical hurdle for clinical translation. Controlled release systems are crucial for delivering growth factors in a way that mimics natural development. Biomaterial scaffolds can be engineered to release signals gradually, using degradable matrices, heparin-based systems, or advanced 3D printing techniques. These technologies help avoid burst release, preserve bioactivity, and allow for the spatial and temporal control needed for complex tissue patterning. The ultimate goal of regenerative dentistry is to grow a complete, functional tooth that erupts and integrates with the jaw. Bioengineered tooth germs, created from epithelial and mesenchymal cells, have shown success in animal models, forming roots, nerves, and blood vessels. However, scaling this approach to humans requires precise control of signaling, cell identity, and developmental timing. Organoid technology and protein inhibition strategies are also being explored to trigger natural tooth growth. Current regenerative treatments focus on repairing or replacing specific dental tissues. Pulp dentin regeneration uses stem cells and scaffolds to restore tooth vitality. Periodontal regeneration targets the supporting structures lost to gum disease. Bone grafting and augmentation use bioactive materials to rebuild the jaw before implants. Emerging technologies, such as gene therapy and 3D bioprinting, promise more personalized and effective solutions in the near future. Biomaterials serve as the temporary extracellular matrix in dental tissue engineering. Natural polymers like collagen and chitosan offer biocompatibility, while synthetic polymers provide tunable mechanical properties. Bioceramics and bioactive glasses mimic the mineral phase of enamel and dentin, supporting mineralization and integration. Multiphasic scaffolds are designed for specific tissues, such as pulp, dentin, or periodontal ligament, and advanced bioinks enable 3D bioprinting of complex structures. Despite remarkable progress, tooth regeneration faces significant challenges. Replicating the highly organized structure of enamel and dentin is a major material science hurdle. Sourcing functional, non-senescent stem cells is difficult, especially in older patients. Achieving rapid vascularization and innervation, controlling morphogenetic signaling, and ensuring immune compatibility are ongoing obstacles. Translating animal model success to human clinical practice remains a complex task. Safety and ethics are central to the clinical translation of tooth regeneration. Pluripotent stem cells carry a risk of tumor formation, and allogenic therapies may trigger immune rejection. Achieving correct tooth shape and integration is technically demanding. Ethically, stem cell sourcing should avoid embryonic tissues when possible, and patients must be fully informed about the experimental nature and risks of these treatments. Accessibility and cost are also important considerations for equitable healthcare. Cutting-edge technologies are accelerating the field. Gene editing tools like CRISPR, exosome-based therapies, and protein inhibition strategies are being developed to enhance regeneration and reduce risks. 3D bioprinting allows for the creation of patient-specific scaffolds and tissues. These advances promise quicker recovery, more personalized treatments, and a shift toward biological rather than mechanical dental care. By 2030, stem cell-based enamel repair may become routine in dental offices, reducing the need for fillings and crowns. By 2040, lab-grown teeth could become a viable alternative to implants, offering a biological solution that feels and functions like natural teeth. Advances in bone regeneration will mean fewer surgeries and faster recovery for patients with gum disease. The broader impact will be a shift from damage control to true restoration in dental care. Tooth regeneration stands at the intersection of biology, engineering, and clinical practice. While whole tooth regeneration in humans remains a challenge, advances in stem cell science, biomaterials, and signaling control are paving the way for meaningful clinical therapies. Ethical, safety, and regulatory considerations must guide progress. With interdisciplinary collaboration and continued innovation, the vision of regrowing natural functional teeth is moving closer to reality.